Hello everyone and welcome to the Dendrite Podcast. My name is Farnham and I'm here with my co-host Said. Dendrite Podcast is a part of the Dendrite Club at the University of Ottawa, where we focus on making scientific literature more approachable and helping students get involved in research, whether they're just starting out or looking to deepen their understanding in a specific field. In each episode, we sit down with a professor or researcher to discuss their area of interest in research and their life in academia. Each month, our episodes are followed by an academic journal club meeting where we explore the topics of our episodes and take a broader look at interesting recent research in the theme of the month.
Today, we have Dr. Baptiste Lacoste as our guest. He's an associate professor at the University of Ottawa's Faculty of Medicine and a principal investigator at the Brain and Mind Research Institute. His work centers around neurovascular biology, examining how brain's blood vessels support brain function and what happens when they are compromised from an early age. His lab explores neurovascular development, neurovascular injuries, and how neurovascular interactions play a role in neurodevelopmental disorders, particularly in autism. So, without further ado, let's begin this conversation.
Awesome. I'm very excited. First of all, let's say hello to Dr. Lacoste. How you doing?
I'm great, fine, and thank you for having me on the podcast.
Perfect. Thank you very much for coming and be as a guest of our podcast. I know you had a very busy schedule, and I know you have a seminar coming up in a week or so, right?
Yeah, preparing for a nice trip.
Okay, cool. Awesome. Well, thank you very much for being here today. I just want to explore a little bit of the topics that you are working in your lab. Just give us a brief introduction to it, and what have you been working so far in your lab?
Sure. Thank you for this question. So, if I summarize, we have three main themes in the lab, all around that what you just mentioned, which is neurovascular biology. The first thing is to know how vessels in the brain develop, so how they form, before even knowing how they get compromised. Mm-hmm. This involves research on glial biology because we look at astrocytes and their control of vascular development. Then the second big theme of the lab is neurovascular injury. So we have studies on stroke, as you might have seen, and ischemic injury. And I would say the third but main theme of the lab is mixing this idea of vascular development with neurological disorders and basically looking at neurovascular interactions in neurodevelopmental conditions such as autism. So these are the big avenues we have developed in the lab.
Perfect. That is very interesting. So you mentioned disorders. What kind of disorders specifically are we talking about here?
Yeah, so the lab, since it started, so we started in 2016, has really taken a full spin on neurodevelopmental conditions. So I know it's a kind of a vague word or expression. It englobes autism per se, but we talk more about autism spectrum disorders because it's a wide range of conditions. We also have expertise on Down syndrome and another syndrome we call Fragile X syndrome. And more recently, we have obtained funding to look at the SYNGAP1 deletion syndrome, which is another type of intellectual disability.
Right.
So you see that it's a vast amount of disorders, but we classify them in neurodevelopmental conditions.
So I just wanted to ask, because you've had an incredible academic journey from University of Bordeaux to then Montreal, McGill, Harvard, then University of Ottawa. When was the first time that you became interested in neurovascular interactions? How did the whole journey start?
That's an interesting question. Very late. So when I was in Bordeaux, as a young student, I was doing my undergrad studies and mostly interested in neuroscience. So I was fascinated by neurobiology. And I wanted to know how a synapse works, how neurons communicate. So this really was a passion. And then I started to do a master's study in Bordeaux based on Parkinson's disease. So it was neuroinflammation and Parkinson's disease. And then I wanted to know more about, diseases of the brain. And one of the things that young students should do is get away from their comfort zone and country if they can, if it is affordable. And I moved to Canada to do a PhD in a lab of a now passed away researcher who had a specialty in electron microscopy. And he was studying the underpinnings of depression and mood disorders in terms of peptide trafficking of different receptors, which has nothing to do with the blood vessels. So as you can see, I have a neuroscience background. I am more of a neuroanatomist. Mm-hmm. I'm a morphologist rather than even a physiologist. And then when you want to become a professor, a PhD is unfortunately not enough, and you need to do a postdoctoral fellowship. Mm-hmm. So I wanted to keep working on diseases. That's what I'm interested in. I had this twist on neuroinflammation. And I moved to the lab of Dr. Edith Hamel at McGill University at the Montreal Neurological Institute. And she was working on Alzheimer's disease and neuroinflammation. It was perfect for me. So looking at glia and neuronal defects. But she also had a twist on the vasculature. And she was looking at blood flow dysfunction in Alzheimer mouse models. And in that lab, I said, oh, well, this is very interesting. So I spent almost 3 years there. Mm-hmm. But that was not enough. When I saw all these vessels, degrading and losing their function, I asked myself, well, what about the other extreme of life? How do they develop from the get-go? So I sent a few emails randomly, and the first person who answered me was a PI from Harvard University, Chenghua Gu, who at the time was a younger scientist looking for postdocs. And she said she answered within 15 minutes saying, oh, yeah, I'm interested. So long story short, I went for an interview, made it, and then spent 3.5 years looking at how vessels develop.
Wow.
Okay. So then I come back and say, well, I'm going to do what I want. And I took the disease, vasculature, development, and I decided to work on autism.
Awesome. That's very cool. Well, I'm sure throughout the whole experience, you had different passions and different interests. What is it that is like currently is your main passion, and you're trying to like find the solution or find the answer for that question specifically? What is it that keep you, I guess, awake in nights and mornings, I would say?
Oh, yeah. At night for sure. Because it becomes an obsession when you're really passionate.
Yeah. So what is it that you're currently really interested in and trying to find an answer for?
So that's an excellent question. And the word passion is actually well chosen, because you cannot be so much invested and committed in something you're not passionate for. Mm-hmm. So my quest, I'm going to call it this way, is finding how you could treat autism by treating the vascular defects. And the reason why it becomes more and more every day a passion is because we are finding the causes now of vascular deficits in autism, at least in mouse and cellular models. And recently, which is unpublished, we actually started to find a way to correct these deficits. So we do now think about applications in the clinic. We are, in the steps of filing patents and wanting to move into the next steps. So that's my passion, is really helping patients and their family that suffer from autism from an, out of the box thinking perspective, which is through the vascular system.
That is awesome. We just want to maybe dig deeper a little bit. What kind of techniques you are using in this particular research area, and give us a more, like, if you want to put it in simple words, what is it that you are actually doing to find those patterns of autisms?
That's a great question. So we might not have time to describe what are the models we use and how they were generated and how we model autism in mice. This is a long story, but I can tell you we do a lot of things. So my lab, I would say our lab, because it's a teamwork, it's not my thing, we work as a team. We are very multidisciplinary. So it's not one technique, it's many techniques, from physiology to imaging to molecular measurement. So we do a lot of macroscopy, we do a lot of electron microscopy, so a very specific way to image. We do mouse behavior, we do RNA, sequencing, we do measure metabolites, lipids, in cells, in plasma samples. So we do almost the whole spectrum of experiments you can think of, from in vitro to in vivo, in laboratory models. And this is helping us to, go deep in the science. That's what I want. I want an holistic view of a disease, but also a very thorough analysis at every level. So that basically that's the philosophy we have.
So how do you make the connections between all these different research areas? So there must be this translation between the behavior of the mouse model and then what we see in vitro, and then from mouse to humans, obviously. So basically the link between all this fascinating different areas that you were talking about.
That's a fantastic question, because this is our job, is making links. So usually it depends. My lab is a bit special, and I am a bit special. So we start by very exploratory approaches, saying, and even betting, oh, we're gonna find something. So let's do all these things. So we started by exploring and finding, and being sometimes lucky to find one path, and then we follow that path very deeply and with a lot of intensity. So when I say RNA sequencing can lead us to a drug target, it's actually what's happening. Is you find some changes at the molecular level, and you say, oh, that's interesting, let's follow up that track. Then you try to identify what molecule is probably changed, and you say, well, oh, that's a molecule that is found at a lower level that it should be in a normal situation. And then you say, oh, well, what about we bring back that molecule? So we do what we call a rescue experiment, and then we bring back some molecules, or we stimulate some pathways. And you're like, oh, it works in cells. Let's try to do it in a slice, and then let's try to do it in a brain organoid, and let's try to do it in mice. So we are now at the level where we can say, by bringing back so I can even tell you, because this is common knowledge now, but we can bring back some energy levels in the cells and make them function better, the vascular cells actually. And then now the next step for us is trying in mice, and then look at behavior. So if we can correct an autism-like behavior in mice using a molecule, when you say, well, we're gonna try this in humans next, of course it goes through ethics and then legal issues and many things, but that's the path.
That's awesome, and we really look forward to a day that we can actually have those experiments in humans. Well, one specific key topics of your whole research and experience is blood-brain barriers. And from what I understand is basically it's a security system for our brain to see what goes inside and outside. And basically from what I understood is it's keeping some molecules or some atoms outside and some inside. So I'm just curious if you can maybe explain a little bit more about what blood-brain barriers is.
Yeah, sure. So it's a fascinating question and topic, extremely big and comprehensive. So again, long story short, the vessels of the brain are very special. So all what I told you about autism and our work is not really based on blood-brain barrier, but the blood-brain barrier. I'm going to call it BBB because I'm French. It's very hard to pronounce.
Yeah, I know.
So the BBB is associated with many conditions and many interesting phenomena. So the vessels of the brain are very special in the way that, as opposed to vessels from kidney or liver or even the skin, they don't leak. Nothing can go through a vascular tree in the brain. Why? Because they have very specific properties that make them sealed. You have very specific junctions. You have very specific pathways. You have purposely lack of communication or lack of transport that makes these vessels a barrier. So the blood to brain barrier is made such as you said, toxic molecules won't go in the brain because let's say you eat something toxic, you are exposed in your environment to something toxic. It's going to go in your bloodstream. The brain is protected against that. You have an inflammation, you have bacteria. It's protected against that thanks to the blood vasculature, the brain vasculature. The liver, the vessels are in the liver, kidney, the vessels are completely open. Everything that is in the blood will go in the organ. You know what I mean? The brain is very special in this way. So to go to disease, I can very shortly tell you multiple sclerosis, MS, is characterized in its first steps by an opening of the blood-brain barrier to immune cells that are going to go in the brain and destroy synapses and neurons, so which is causing motor symptoms. Okay, Alzheimer's disease, one of the later stages of the disease, is an opening of the BBB, again causing neuronal death through incoming of all these molecules that should not be in the brain. People are suggesting that autism is linked to BBB defects. We are on this path too. It's very preliminary, but that's an opening field. So that's a concept that people are following up on. Yeah.
So can we now speak about endothelial cells in particular, and what is the developmental aspects that might cause some defects later on that would link to autism or any other disorder?
It's an interesting question. That's exactly what we're actually working on. If I summarize what our lab is finding currently is these endothelial cells. So endothelium means an epithelium inside. Endo means an inside, and thelium comes from epithelium. So it's like a very thin single layer of cells that form the tubing of these vessels. And many people think that this is like a tubing. We are like the plumbers of the brain, and then we fix tubes. But these endothelial cells not only form a sealed tube, but also secrete a lot of things. They secrete growth factor that can help neuronal development. So again, I could spend hours on this. So what we are currently finding in the lab is those cells don't function properly. They have a lack of energy that do not prevent them from being alive. So your vessels don't collapse. You don't have hemorrhages in the brain, but they don't support the functions that are triggered by different stimuli. One of them is cerebral blood flow. We can talk about this after if you want, but when neuronal activity is increased, usually you have a corresponding increase in blood flow. The regulation of this process through neurotransmitter release and other cells implies endothelial integrity. The endothelium needs to be functioning properly. So we are finding that this endothelium does not work properly. And we are finding that correcting this dysfunction helps in autism, at least in mice and cells.
Right.
So then how do you screen for some maybe proteins or genes that regulate the activities of these cells, or then maybe they might cause these cells to have defects? So like ROC2 maybe.
Yeah, so ROC2 is one of the things we worked on recently on a small paper we published recently. Even before ROC2, I can go back to the ROC study. The way we really discovered that dysfunction of cells is having some screening tools in which you can isolate these cells from a brain, mouse brain, and then you have a suspensions of pure endothelial cells that you either look at gene expression or a protein expression and metabolite content, but also function in different assays. So we've seen that they have less of energy molecules such as ATP, adenosine triphosphate. They possess less of this, so they have less energy. They function better when we bring back ATP. So that's something we are currently working on. It's subject to an actually patent application, and we disclose it in different seminars I give, so it's not a secret. About the ROC2 involvement, it's very interesting because the particular mutation we are focusing on in the lab, which is a mutation commonly associated with autism in humans, causes an increase in ROC2 activity, which is kind of, I'm going to say it in a simplistic way, toxic to the cells, and particularly the endothelial cells. When you reduce ROC2 expression and activity, it's good for the system, and it's good for the endothelium. So what we did recently is take a mouse model of autism and cross it with another mouse that has low ROC2 expression, and it helped many things. Behavior, so we could, by reducing ROC2 function, ROC2 is an isoform of ROC, enzyme. By reducing ROC2 levels, we could not fix but improve a lot of things in this mouse model. Vascular function and behavior in mice.
Speaking of improving the vascular function, I just want to explore more of the effects that would change that vascular function. I mean, you earlier mentioned about the plasticity of this whole system. So I'm just wondering if there's effects like aging or I really love the there's a phrase called use it or lose it, but I always thought it was more related to the neurons. But now I'm seeing that there's also another whole word about the vascular part of it also.
Absolutely.
So why don't you let us more about the things that can affect in case of the vascular aspect of it, not the neurons itself?
Well, it's a very interesting question, and it starts from birth. So actually, when I was at Harvard University, my theme of research was how sensory input, so environmental sensory input, shape brain vasculature. So what I discovered at this time is if you remove sensory input and even input from the skin, so at the time we were working with whiskers in mice. If you remove the whiskers in mice and then you start to just wait and see how the vasculature develops, it develops less in the region that is associated with that specific part of the body. And we had different ways to cut this input. So so skin sensation is going to go to your spinal cord, is going to go to your brainstem, thalamus and cortex. We had different ways to interact with this pathway. And when we cut input from different places, we could see that the vasculature was reduced. So as you say, vascular system in the brain is plastic. It also depends on stimulation. So you can already think that a young kid that lacks environmental stimulation will have a less developed vascular system, which has long-term implication for neuronal function because the vascular system supports vascular development and function on the long term. And then to answer kind of also your question on the other side of life, the plasticity goes the other way. I would say it's not really like if you don't use the vasculature, it disappears, but actually it needs maintenance. The blood-brain barrier needs maintenance. And if you see diseases like Parkinson's, MS, Alzheimer's disease, the aging of neuronal and glial systems affect the way the vasculature is maintained, and then you can lose vascular integrity. If you lose blood flow, less blood is coming. So we didn't touch upon this yet, but blood flow is the only way to bring oxygen to the brain. Blood flow is the only way to bring glucose to the brain, energy, food. So if you lose that, you lose neuronal function. So it's a vicious cycle that is unfortunately feeding itself toward neuronal demise.
Perfect. Well, there you go, folks. You heard it from Dr. Lacoste himself. You gotta really challenge yourself, and as he mentioned earlier, try to really come out of your comfort zone to be able to maintain those vascular functions.
So, what therapeutic pathways can we now think about? So, how can we maintain this the health of this vascular neurovascular system on both ends of developmental aspect of, so at the beginning of the life, the end of the life, and throughout. So what can we do about it?
Well, we just want to maybe know some practical advice for people who are getting older these days, with social media, ChatGPT, we don't really get to use our brain. So what are some practical advice that you can give us?
I'll answer both questions at once because and then I think both of what you just touched upon is very interesting. You have drugs that can help, and you have behaviors that can help.
Exactly.
So we are working on future drugs that can help to regenerate endothelial function, to support long-term neuronal function, because we know now there is a crosstalk between these two. People are working on this actively. You can maintain BBB, blood-brain barrier integrity, through different molecules, and I'm not going to list them. This is not my exact field of expertise, but many companies invest billions of dollars in fixing the BBB or going through it, because you need to go through the wall to deliver drugs to the brain. And unfortunately, the BBB is a limitation for drug delivery. So this is a huge field of research, and we are actively involved in autism field. So give us 10 years, we'll give you an answer. Concerning the behavior, I would say not to kill all the companies that invest all the billion of dollars. One of the things that is so easy to do to target your blood vessel in a good way is exercise and a good diet. So good and good practices in just life, diet, exercise, not becoming sedentary, moving. Some people cannot do exercise. All older people or people with handicaps cannot go to the gym, but at least they can move a little bit. a constant movement will trigger a lot of things in your brain, including secretion of different molecules that will fix your blood vessels. And the blood vessels are the first line of defense for the brain. So a lot of scientists now agree that even from early life, constant exercise and then a mix with a healthy diet is going to be helping your blood vessels, hence helping neuronal health. That's a common knowledge now. And then books, you can read many books about this. You can read science emerging in this field, which is very convincing. People have done it in mice, in rats, but now in humans, we have the proof that as simple as exercise and a good Greek diet is very good for your blood vessels.
And then what are the things to be worried about, like in specific maybe periods of time? So for example, I don't know, something happens like prenatal hypoxia now, and other things. So what are the dangers that this endothelial cells or in general neurovascular system is prone to?
Yeah, absolutely. I mean, that's something we work with Dr. Thébault, who's at the Ottawa hospital on this theme. Perinatal injury. So you have to understand one thing. So the blood vasculature, the brain vasculature is very sensitive to injury. And the consequences are long-lasting. So if you have, during these critical plasticity windows, this is the word we use, an impact on the brain vasculature, it's going to give long-lasting neuronal consequences and behavioral and autism, for instance. Hyperoxia, so too much oxygen at birth, has been shown by Dr. Thébault recently in collaboration with us to drive cerebral vascular changes, reduce blood flow, and then long-term neuronal deficits. And in mice, it actually triggers behavior that are reminiscent of autism. Hypoxia, hypoxia is a classic injury around birth. You might have heard about cerebral palsy. If you have a perinatal injury, such as a perinatal ischemic event, it leads to death of neurons in certain regions of the brain and cerebral palsy. And people have recently linked perinatal hypoxia to autism. So you see the long-term consequences when you hit that critical window of plasticity perinatally. So that's exactly what we should think of, is if you know a target in the vascular system, well, treat early to help or to prevent a course of things that could happen during life.
Now we can talk about the interactions between neurovascular system and neurotransmitters. So like serotonin, dopamine. What is the interaction here, and how does it affect depression or other brain disorders?
Yeah, that's an excellent question, and so it's a big field of research, and I can start by telling you, for instance, that those neurotransmitters you might know, which are very classic. If you see serotonin, it reminds you different brain function, but also depression, because we know that serotonin levels are low in depression. Dopamine, you might relate to the effect of, let's say, some psychostimulants such as cocaine, which is a blocker of dopamine reuptake to enhance the effect of dopamine. Adrenaline or noradrenaline or norepinephrine, which is the same thing, is a drug released for an action of defense, and this is affecting neuronal system, but also vascular. So serotonin is a potent vasoconstrictor. Serotonin is going to close your blood vessels. Noradrenaline is going to also close the blood vessels. Dopamine is less studied, but these are responses which are adaptative responses. And then these are not just neurotransmitters. They actually act on different pathways, including directly on the endothelial cells. Acetylcholine. You might have learned in your course long time ago that acetylcholine drives muscle contraction through the neuromuscular junction. Well, acetylcholine does the opposite effect on the vasculature of the brain. It's a vasodilator. So it's going to open up the blood vessels. And you know that acetylcholine is usually secreted when you need arousal. It comes from a deep region of the brain, the basal forebrain nuclei, and it projects onto the cortex here for dilating blood vessels. In the same time, it regulates neuronal activity. So yes, neurotransmitters have a direct effect on vascular function in the brain. And just to finish, serotonin is low. You can imagine in depression, that it will affect the blood flow in the brain. In Alzheimer's disease, one of the first systems that is disappearing is the acetylcholinergic system. So you can imagine that it has an effect on blood flow as well. And it is known that all those diseases have a vascular component. So that's a huge field of research.
Now, just to have a general question about how big is the research community talking or investigating about neurovascular systems? Because, well, thinking about autism or depression, we usually just think about neurons and then neurotransmitters. But this other component of it, which makes incredible sense, because you want the neurons to have oxygen, to have glucose, and it's by the blood flow. So how big is this area of research?
Yeah, so if you would just keep the area of neurovascular interaction field, it's big. You have lots of people working on neurodegenerative disease. So lots of people working on Alzheimer's disease, for instance, in human and animals and laboratory models. You have lots of people working on Parkinson's. Recently, the field of depression and its relation to the vasculature developed. And we actually have one famous scientist in Canada, Caroline Ménard, in Quebec City, who has developed her lab around BBB regulation in depression, and she finds very interesting targets. Concerning autism and vascular, well, we are the only lab. So it's a small field, and I'm very happy to be developing this niche. But otherwise, the field of cerebrovascular biology is pretty big.
Awesome. So let's switch the gears a little bit and ask Dr. Lacoste, what do you do outside of the lab? Is there any hobbies that you're trying to unwind from this complexity of neurons and neurovasculars?
I do a lot of things. So first and foremost, I am a husband and I'm a father. So I have a cute little family, which I'm very happy to get back to every night. But that's not a hobby. In terms of side hobbies, so I do a lot of photography. So I love art, and I actually think it helps me in my science, because illustration and demonstration have to be so thorough and convincing to an audience that if you have an artistic twist to it, it's going to be helping you a lot. So I love photography, and that helps me in the imaging we do in the lab. And I also do a lot of astrophotography, which is the complete opposite of what I do in a microscope. So I love extremes. I looked at molecules, but I also look at nebula, which is something I do in my backyard. And I play piano, and I play the guitar, and I read a lot of books. So yes, I have a life outside the lab.
Well, it's a big sphere of hobbies. Now I was wondering if you could talk to us about a time that you have faced failure, maybe in your career or just in academia in general, and how did you cope with it, and how did you step up again, and what did you learn from it?
Okay, I could spend hours on this, so let's stop me. So I kept on facing failures. So I come from being first of all, I have a pronounced attention deficit. I have been diagnosed with dyslexia. I am dysorthographic. I have a lot of issues. So, and I'm French. So it's a huge phenotype. But more seriously, I was a very, very average student. I had difficulty concentrating. I had to redo one of my secondary classes to actually retake off. So that was a big failure, but that helped me a lot because I could reset the clock, and it actually helped me a lot. I saw specialists to help with my dyslexia, and I could have been treated for attention deficit, but I did not at the time. But it was a long battle against myself. And when I discovered my passion for neuroscience, it was an awakening that something that people should know is the field of neuroscience and research in neurobiology, probably other fields as well. Maybe less astrophysics. You don't need much math. You don't need much chemistry. You don't need much expertise when you start, because you learn everything on site, on the go, at the bench, with your mentor. So all the things I know today, I've learned them after graduating, school and after all these diplomas that you should have. So failure is my history. and then nowadays, well, as you might know, to keep a lab working and alive, you need to find funds, and we keep on writing grant applications, and we keep on failing because it's very competitive. And to find one, let's say, CIHR grant to fund your lab for 5 years, you have to fail maybe 3 to 4 times before getting one of those big grants. So failure is part of the game. Failure is part of science, and you can only learn from failure. Successes are good, delightful. You celebrate, and the top of the wave is very agreeable. But when you are at top of a wave, if you surf, you know that you go down very fast too. So it's a roller coaster, and then people should know that science is up and downs, but the downs are also the way you take off, and the taking off to me is the best part. The top of the wave is I know something else is coming, but the climbing phase is amazing. But you have to start from low, right? So please continue to fail. That's my message.
And can I ask you, if you were not a neuroscientist, what career would you choose?
That's a good question. People like me with an attention deficit and hyperactivity, they think they could be so many things at once, and it's hard to choose. When I was young, so let's say when I was 18, I wanted to be a rock star. I wanted to play the guitar in front of a big stadium. I had this. When I was younger, I wanted to be a fireman. Yeah. No, but more seriously, I wanted to write books. So I could have been a writer, and I think I could have been a musician.
And you have followed on those interests
as hobby. Hobbies. I didn't give up, but I kept them as hobby. And I must say, I had very good advices from my parents. They say, don't make your hobby your job, because once it becomes your job, you lose it as a hobby, and that's very sad. You have to keep your hobbies. And then there was a wise choice.
Well, that's great. And another advice from my parents is always trying to keep the balance between your hobbies and your career.
Yes.
And if you want to make connections, I guess you could say the security system of our brain, blood-brain barriers, they're always trying to keep that balance. So
absolutely,
we could definitely consider that.
That's a good comparison.
So now we can maybe talk about teaching as well. So last year you won the Excellence in Teaching Award in Faculty of Medicine. How do you approach teaching in general? What is your relationship with the students?
Yeah. It's so fortunately to me, this is something I think I have in the blood. So what I mean by this is I don't really force myself to be approachable. So the first thing I do is telling the students I don't care about the details. And I always say when I teach, on each slide I present, we don't really pay attention to the names of the molecules, to the structures of the molecules, but what I want you to learn are the concepts. Because if you learn and understand the concept, this is what you will remember. Even a glimpse of it 10 years later. But I wish I had teachers that would say, don't learn all this by heart. Don't spit all on the final exam and then forget the next week.
Teachers like yourself.
Exactly. So I basically teach the way I would have loved to be taught.
So then what advice would you have for students who are trying to just understand what is research, or trying to approach this scientific mindset?
Yeah, because as an undergrad, we always struggle to find the right prof. And as you already know, and our audience know for sure, that we have to send 50 emails, 100 emails, and then maybe 2 or 3 profs get back to us. So I'm just wondering if undergrad students specifically want to be involved in your research, what skills or knowledge they have to work on, and how should they approach you in case they want to be involved in your research?
Okay, well, so the answer is quite simple, but at the same time, it might be complex for some people. I don't look for any skill apart from the people skill. So, and then, fortunately, it's a good problem to have. Unfortunately for students, my lab is rapidly at high capacity, and usually when I answer. So I will always answer requests. These days I answer no very often because we're full. So that's the only problem we have. And I tell you, it is a good problem to have. But the only thing I'm looking at is the inner drive. When I meet a person in my office for like, I'm not evaluating anything else than, is this person genuinely interested in what we do? That's a bonus, I would say. If you come for a good reason, it's normal. It's the game. Everybody applies to 15 labs, 20, 30 labs. Mm-hmm. You are going to craft your letter to the lab you apply to. That's the game. That's normal. You would do it for any job. Mm-hmm. But the day we meet, if I feel you actually have a genuine interest in neurobiology of disease, you don't have to have an interest in vasculature. We will drive this interest in you. It has to come from the inside. I cannot implement in someone the flame for science. I can help someone stay on track. The way I mentor even my lab members is I guide people to stay on track, to avoid hitting too many walls. Yet, I think it's good to hit a wall from time to time, but I want it to be not too much of a huge crash. So I put an airbag in between. That's my job. But the only advice I have to people is be yourself. Don't use ChatGPT to write your letters of motivation, because I'm going to even tell you, sometimes I take a letter which is so well, perfectly written, and I go to ChatGPT and I say, what are the chances you wrote that letter? And ChatGPT says there is 95% chances that I wrote that letter. So you won't fool me. A letter which is very personal, which has typos, which has mistakes, but is conveying that passion or that will to discover. That's going to be the letter I'm going to be interested in and following up with the student. Okay, so be yourself and don't go meet with someone that you are not 100% sure you're interested in. As a student, don't reach out to people you say, I need to find a position, I have no choice, I go to this person, but it's not really my passion or interest. Don't even bother applying, because we will feel that lack of connection.
Well, I'm sure the folks really enjoyed the last part of trying to be yourself and know yourself. I just want to, maybe it's going to be a little bit philosophical, but just wondering, what is the purpose of your life? You said you're passionate about neuroscience and basically creating a great life for your kids and all. But what is it that you as a person personally looking forward to achieve in your own life?
Not to get the Nobel Prize, that's for sure. Provide a meaningful contribution,
right, to
the society. I mean, we are here for, let's be lucky, 80 years, 90, if you do exercise. To leave a trace, but more than this, to have a meaningful contribution to our society, while not impacting our planet, too badly, because planet doesn't care about us.
Right.
She doesn't want us. so let's have a symbiotic relationship with the Earth, and while contributing to the society in a meaningful way. That's the main goal of my life.
Perfect. That's awesome.
Dr. Lacoste, it was a fascinating conversation. Thank you for being a guest in our podcast.
Thank you for the invite and fascinating discussion.
Thank you for listening to our conversation with Dr. Lacoste. In this episode, we discussed neurovascular development, injuries, and neurodevelopmental disorders, particularly autism. We also explored the techniques Dr. Lacoste uses in his lab and their potential clinical applications.
Dr. Lacoste also shared his personal story and a glimpse into his academic journey and perspective on life. We added a link to his website in case you're curious and want to learn more about his work.
This is a Dendrite podcast. To support it, subscribe and share this episode with your friends and anyone who you think might be interested. We will discuss research in neurovascular systems further in our monthly academic journal club, so make sure to be on the lookout for the meeting as well. Thank you again for listening to us, and we hope to see you next time.